This is the Unit 5.1 video on gene regulation in prokaryotes. So to begin with, you might not have an idea of what I mean by gene regulation, and we'll get to the prokaryotes part later. But first of all, let's focus on just the gene regulation. Let's start with a question. The question is, if all of our cells have the same DNA in them, which they do, all of our cells have all the same DNA, all the same chromosomes, all the same genes, how do they have different functions? So this first picture here is a neuron or a nerve cell. Now think about the things that your nerve cells do. They send messages to your brain and then back from your brain to your muscles. So they have a very specific job. What they do is very different from, for example, the cells of your pancreas, which are right here. Now the pancreas cells produce insulin, which helps you in the digestion of sugar. They do not transmit messages the same way the neurons do. So how is that possible if they have the same DNA? And then finally, here's another very common type of cell we're aware of, muscle cells. Muscle cells have a very specific job. They contract and relax to help you exert force. Neuron cells don't do that. Pancreas cells don't do that. So how does that work if they all have the same DNA? Well, what it comes down to is that genes and thereby pieces of DNA or RNA can be turned off or turned on depending on what cell they're in and depending on when they're needed. So the neuron does not have all of its genes turned on. It doesn't need the same genes that the pancreas cells or the muscle cells would. So those pancreas and muscle cell genes are turned off in the neuron cells. And of course the same goes for the pancreas cells and the muscle cells. Genes are turned off in them that they don't need. So we're going to talk a little bit more about that, what that means to turn on or off a gene. All right, before we get into that into great detail, we need to talk about what's called the central dogma of biology. By central dogma, it just means like kind of the big important idea of biology. And you guys are already aware of this. Whether or not you've called it the central dogma doesn't matter. We've talked about this in class. Number one, we have DNA replication. So DNA polymerase helps to make a copy of DNA. From there, RNA polymerase can transcribe that DNA and turn it into RNA. And then third, we can have translation occur to turn that RNA into a protein. So the sequence DNA to RNA to protein is kind of the main idea that everything in biology is built upon. So when we look at that central dogma of biology, the question we're going to be asking in today's video is, where could that regulation occur? What steps during this process from DNA to RNA to protein could we have genes turned off and turned on? In other words, could we have their products, the proteins, made or not made, depending on what's happening in those steps? Well, the first step, replication, doesn't really matter as far as gene regulation goes, because we always have multiple copies of all the DNA. So replication is not a place where we're going to regulate or control when genes are turned off and turned on. But transcription and translation are definitely places where we can control what genes are being expressed. And the most common of these two is transcription. So most of the regulation of DNA, essentially done through RNA, is via transcription. So let's just consider here what the normal pathway would look like and then what different regulations would look like. So normally, as we just said in the central dogma, we start with a gene that's on our DNA. The gene gets transcribed or turned into mRNA. That mRNA gets translated into an enzyme, which remember is our protein. And then finally that enzyme produces a product. And that product can do a variety of things in your body. So just a reminder, this gene is the DNA. Gets changed into messenger RNA via transcription. The enzyme is our protein, which gets made via translation. And then finally some end product. So if a gene is working, if it's running, if it's doing what it needs to do, this is a very common scenario for what it might do. It might make an enzyme that creates a product. Now, if we make some changes to that pathway or we add some regulation to that pathway, we could do a variety of things. The first thing we could do is we could inactivate the enzyme. So inactive enzyme. And we've talked about already how we might inactivate an enzyme in class. Remember we talked about enzyme inhibitors? So this can affect whether or not that product is made. So that is one way to regulate proteins. The second way is if we somehow stop translation. So if translation is no longer occurring. If we don't have translation, we don't get a protein, therefore that function doesn't happen in that cell. Finally, we could affect transcription. So making the mRNA, no transcription means no RNA, which means no protein in the long run. All right, so those are three possible ways that we could control what genes are turned on or off in the cell. We'll talk about all of those in more detail in the next video. But for this video, to keep it short and sweet, we're just going to focus on one type of regulation and in one type of cell. So back to that word prokaryotes. So prokaryotes are single-celled creatures like bacteria. And bacteria tend to be simpler to understand when it comes to their genetics compared to eukaryotic cells like ours. So that's why we're going to start with regulation by talking about simple bacteria. The pathway that you're seeing here in this diagram of regulation is the first regulation pathway or the first process for regulating a gene expression that scientists figured out. So it's a kind of famous one, and it is called the lac operon. And why it's called that will hopefully become clear in a little bit. So we're going to look at how a bacteria regulates some of its genes, so turns them on and off, in this area of the genome of the DNA called the lac operon. So this whole stretch of DNA here is the lac operon. And within that lac operon, there are three genes. There's gene 1, gene 2, and gene 3. And they may or may not be transcribed into mRNA, which is this one right here, depending on what kind of regulation is happening. Okay, so that kind of gives you a tour of what's going on. This little egg-shaped thing should look familiar. That's our RNA polymerase. Okay, with that said, how do we turn off and on the transcription of this gene? So this is a transcription example, or these three genes. So in order for transcription to take place, remember this RNA polymerase has to cruise down the DNA, and as it does, it's going to copy one side of the DNA and turn it into the mRNA. So what happens is bacteria in normal circumstances, in its normal environment, has these red repressors around, or inhibitors. Okay, and as you can see, these inhibitors fit right there. So that red inhibitor, if it's around, it's going to bind right in here, and it's going to block the RNA polymerase from moving downstream. So you won't get any transcription at all. So transcription is off. So when you have an inhibitor, there's no transcription. Okay, so in other words, we don't get the product eventually of that gene. No protein is made because no mRNA is made. However, sometimes bacteria have in their system, or in their environment, I should say, this little green molecule, which is called lactose. Now maybe lactose sounds familiar. Lactose is the sugar found in milk, and it can be a really good food source for bacteria. And so when lactose is in their environment, they want to be able to use it. They want to be able to digest it and get the energy out of it. And it just so happens that this polypeptide, this protein that's made from that mRNA, helps to break down lactose. So it would be very helpful if, when lactose is in the environment, this polypeptide was made, because then we could use that lactose as a bacteria and get some energy out of it. So how does that work? The lactose binds with the inhibitor. And as you can see, that changes the shape of the inhibitor when the lactose is bound to it. Well, what happens when the shape of the inhibitor is changed? It can't fit into the spot anymore. So now that spot is open. And what does that mean if that spot is open? Now polymerase can cruise down and make a transcript, or make mRNA, from all three of these genes. And if all three of those genes are transcribed to make the mRNA, that means that we can make that protein, that polypeptide, to break down lactose. How convenient. We have lactose in the environment. Now we can finally use those genes so that lactose can be made. So again, so that lactose can be broken down. So as I said, this is the first example of a regulatory process that they found for bacteria. And again, it's turning this gene on and off. So the inhibitor with no transcription means the gene is off. And when lactose is present, we have transcription, and the gene is on. And then again, you make the protein to help break down the lactose so they can get energy. Okay, that's it for this video. For the next video, we'll get into more specific details of how our cells, our eukaryotic cells, regulate genes to turn them on and off as well.